Okay, so this will be the tools and toys part of the presentation, which is actually kind of the way that I got into this business in the first place, is because I had some cool toys that ended up teaching us about marine mammals. So my job today is not just to talk about acoustics, but to try to talk about all the different tools that we have and do sort of an introduction to my co-conspirators in the panel. Now, I have to say, I don't have cool cartoons or chimpanzees that some of the other speakers have had. But one of the things that I think is important when you're thinking about new technology is how the technology is going to answer specific questions or problems that we have. I mean, it's one thing to say, let's make this cool toy, but for its own sake, that's not very meaningful. What's meaningful is to say, here's a question that's a really important question. How can we create some new technology that will help us answer that question? So I think that sort of keeping these questions in mind to start is an important part of the whole approach to new technology. So what is it that we really need to know? Let me see if I can make this guy work here. I'll go to green. So what is it that we really need to know about marine mammals? And in fact, one of the things that is kind of disconcerting when you talk to people in the general public about our understanding of marine mammals, their assumption is that we already know everything there is to know about them. And then, it's hard to get a sense that we don't actually know in detail the geographical distribution. We don't know where the animals go, where they spend their time. We don't actually know, except for with a lot of effort, the real abundance, although we try very hard. In many cases, we don't know the structure of the populations. We don't know how the animals segment themselves in meaningful ways. And this is important, critically important in terms of how we would approach conserving them. We don't know much about behavior. I mean, we see them do things at the surface. There's a lot of underwater behavior that is kind of hidden to us. We know a little bit about the physiology and sort of the uniqueness of it, but there are many more things we could know. And then all of these things help us to figure out what are the threats to their populations going on and impacts from human activity. So these are things that we'd like to do a better job in all of these, and how can new technology help us with that? So I'm very pleased that we have this distinguished panel of experts to help sort out where we are in all this. And just I put them more or less in the order that sort of starting from things that happen kind of in the lab and things that happen sort of with captive animals and people that work in the field. So let's see, maybe we could just, Phil, where are you, Phil? Okay, there's Phil Moore. So Phil Moran works at Southwest Fisheries Science Center, and I assume that genomics has been the focus of his career. And of course, anybody who knows genomics realizes that this is probably the hot field of science for the last decade. This is where things have just exploded. And in God knows, 76 or something, I took a class on it, but probably what I learned is irrelevant at this point, except that I know Watson Crick, and that's about, and everything since then is, this is an incredibly exploding field. So, and then Nick, there you are, Nick works in the same lab, but with a totally different focus, and has to do more with looking at sort of biochemicals in tissues and what we can learn from that. And then Dorian.
He'll be here later.
Okay. Well, we all know and love Dorian. He's been in this business a long time. And the National Marine Mammal Foundation, I'm excited to have him explain that to us. I think of Dorian as being part of the Navy, working with the Navy in the, what do they call it, their marine mammal program in San Diego. And new things that can be done with the Navy captive animals. Greg Shore, Cascadia Research, or Greg? Oh, there you are. Right. And so Cascadia, of course, is a really important player in terms of doing field work in Southern California, and then developing the technology for tagging has really come out of work with Greg, and I guess Rex Andrew would be a collaborator in this in terms of the new technology. Brandon is at UC Santa Cruz, but before this he was at the Office of Protected Resources at NOAA, and SCA is Brandon's company. And Brandon has been a really key player in terms of the regulations for sound exposure, and then sort of working out what it is we need to know to understand the impact of sound. And so he'll be talking about mostly the SoCal 10 behavioral response study and things that have come out of that. And that it really has, it's partly technical development in terms of things like how you operate an acoustic source and tagging, but also putting together a large collaborative group and how that all works. And then I am at SIO and a physicist acoustician by training, and try not to get in too much trouble when I say the word biology. Okay, so now I'm talking about Phil's work, and one of the things that has happened in the last, say, 10 years is the ability to take some genetic material and rapidly make a sequence, and make a sequence on a lot of individuals. And mitochondrial DNA is particularly important because it goes from always on the maternal side. And actually, I learned something about this from my brother, who decided that he wanted to have his mitochondrial DNA sequenced. There was a project for the National Geographic. And my mother, whose name is Farrell, would be the origin of my brother's DNA. And sure enough, she had a Celtic origin in her mitochondrial sequence. So there we go. Where you can have this done to yourself and figure out, your mother's, location of origin. But in this case, the picture here shows different ecotypes of killer whales, and I think these are all in the Antarctic. And the application of this technology to killer whales, and I stared at this slide for a while, and I confirmed something with Phil before I said this or whatever. This is a tree that shows sort of the relatedness of these various stocks of killer whales. And basically, how the tree branches shows you how far away they are one from the other based on sequencing of the mitochondria. And the thing that really struck me when I looked at this is, why are residents up here and transients down there, given that these guys more or less occupy a lot of the same space? And so in fact, I think that'll be part of his talk to tell us what we've learned about sort of the structure of all of these killer whale ecotypes. And so this obviously is very important work in terms of separating out units for conservation. Okay, so now talking about Nick's work, and I love this title where it's Hormones to Harnessing the Biotech Revolution: How We Make the Most of Projectile Biopsies. And there clearly is a biotech revolution going on, and it's nice that the marine mammal community can tie into this. And so this picture, where you start out with your lowly skin biopsy sample, shows all these, I don't know, whatever it is, 7 different things that you can do with it. And tying into contaminants, like in the last talk, all the way up to DNA, like what we were just talking about, or RNA with Phil. And then I think, at least from what I know of Nick's work, there's a lot of development here in terms of what you can do with hormones, in terms of things like stress, or he can tell you if the animal's pregnant, and these various other parameters. So you're way better off learning this from Nick than from me, but this is the teaser to come and listen to Nick. Now, this is from Dorian, and it has to do with the auditory evoked potential, which is a technique basically of testing the hearing of animals, and it's also used on people, but how they made a transition from these techniques where we understand them in people to applying them to marine mammals. And it's the technique basically says, I'm going to make a sound in the ear, I'm going to make a click in the ear, And then we're going to record the brain waves that are associated with that click. And from that, we can learn something about the response of the hearing of the animal. And you can do this, for instance, with newborns, because you can immediately learn if the animal, if the newborn is deaf or not. So here there are stranded animals, and also captive animals from the Navy's group of animals that they work with. And so one of the ideas that you could test species that we wouldn't be able to train for a hearing test. We can test what their hearing is. So some species strands, and you can go up and put on this AEP apparatus and then come up with a hearing curve. Also, if you can do this rapidly, you can do a lot of animals, so you get a population level sort of study of hearing, as opposed to just the one animal that you spent 6 months training. It's portable, field deployable, and I think the push there is to get to where you can give it to the stranding networks or other people who would be involved in response to the stranding, and have them collect the data rather than having the specialist go there. So this has a lot of potential for expanding on understanding of marine mammal hearing. Now, the other thing that Dorian sent to me was the idea of using their sea lions in a new approach to tagging. Now, this is quite innovative. And there's an existing program where the marine mammal captive animals are used to detect and also somehow impede the progress of an underwater swimmer. And you can see how this would be helpful in terms of security, because you could have your sea lion patrolling, and when some unwelcome visitor comes to the side of your ship, then the animal can go and somehow detain them, or at least let you know that they're there. Well, you can extend this concept of the sea lion approaching a swimmer to have the one marine mammal approach another marine mammal. And I take it from this picture. Unfortunately, Dorian isn't here to confirm that, but this is probably one of their captive bottlenose that's being approached by a sea lion to test tagging. So this is a cool idea that has a lot of potential in terms of these guys helping us with tagging marine mammals. So I wanted to say just a little bit about the tag technology, and we all know, if you've watched this over the last decade, that it's really rapidly advanced in mostly in the miniaturization of these things and the ability to have multiple sensors. And I stole these pictures. I think this is sort of the one thing that Greg Shore will talk about, are these tags that give us location using Argos. But I think things have advanced a great deal from when I stole this from the Anders et al. Paper of 2008 in terms of being able to have location and also some other parameters from these. Maybe these are medium duration tags or potentially long duration tags. The other kind of tags are short duration, where they're usually put on with some kind of suction cup. And I use the bioacoustic probe as an example here. And here's tagging with John Kalmakidis of a blue whale just off of La Jolla. That's Mount Soledad in the background there. And these guys, you can have a whole suite of sensors, and you learn a great deal about the dynamics of the animal, the behavior of the animal, and also including the acoustics. So, and the classic for these is in fact the D-tag. And I apologize that this slide probably doesn't capture the latest. I think it's a D-tag 3. Is that right? Yeah. And so this is a D-tag 2. And these have been sort of the basis of a lot of what we've learned about beak whale behavior and especially the echolocation of beak whales. And it was developed by the Navy mostly for that, but they have 2 audio channels where you can actually get a directionality of the outgoing sound or some received sound. And they last for, on order, I don't know, it's a day or so, or 16 hours. And I think the new ones may be more even. They sample it high enough to get the full bandwidth of the echolocation clicks for most small cetaceans, not all. And then there are other sensors like pitch and roll and heading that you can go back and actually reconstruct a track for the animal. So and here's an example of a D-tag being applied. I guess that's probably a beach whale. And so now what Greg will talk about is some satellite tagging that's been very much focused on the Southern California Bight and what we learned from that. And this slide shows where I think most of the tags are applied here. Okay, and then these are fin whale tracks, and you start to get a sense for how fin whales use this space. And this is going to be a really powerful technique to learn about their migration patterns, and then is there a single stock or are there multiple stocks? Where is it, if you look at the aggregate of where they spend their time, how does that relate to the actual habitat in terms of properties of the water or properties of the prey? And so this is a really exciting new thing, and part of it, I think another spin-off of this will be looking at the way that the Navy or some other or shipping uses this space and how that interacts with the way that the fin whales would use the space or other animals that are being tagged. So, and this is something where 5 years ago we couldn't have done it. Now, Brandon will talk about the behavioral response studies, which are just starting up in SoCal. And there's been one field season. I think it's intended that there will be something like a 5-year program. And it's quite a large interdisciplinary research team that's put together to do this work. And here you see this very iconic picture of a couple of tags going on to sperm whale. And there have been 63, just in this one season. I think this must set some kind of record. That 63 tag attachments on 9 different species, 400 hours of tag data, and 28 controlled exposures on these animals. And this is really impressive relative to some of the other situations where this kind of effort could go in and you wouldn't end up with nearly this quantity of data. So I think there'll be spin-offs besides the primary goal, which is learning about how the animals react to sound. I think there'll be a lot of spin-offs in terms of just behavior because of the quantity of data that we're getting out of it. One of the things Brandon wanted to point out about this is, in terms of doing the controlled exposure experiment, is having a very rapidly deployed, easily used sound source. And that was one thing that happened as part of this development. It's about, I don't know, maybe 6 or 7 feet long, and a set of 15 transducers that you can deploy pretty rapidly. A couple of people can do this. So this is a big plus in terms of being able to, once the animal is tagged, pretty rapidly do your controlled exposure experiment. And then I threw in one slide, and I assume Brandon's going to do the whole suite here, but this is showing what the D-tag people call a pseudo track. So I guess that means it's a track that they're not quite sure it's exactly that way, but it's a proxy for where the animal was tagged, and then just a plan view of where it swam, and then a couple of dots in the middle here are the time of the exposure. And you can see there are these various dives, and then during the exposure, the actual level of received sound can be quantified. So here's the dB scale, and you can see it's upwards of sort of 150 dBs, along the bottom part of the track. And then this is the actual recording of the source at the whale. So anyway, so this is pretty exciting new work that Brandon will talk about tomorrow. Okay, so now in the end, it's all about acoustics, right? At least to some of us. So, and here is the blessed harp. And some of you would say, hey, we saw this last year, so this isn't new. But we're always trying to make it better. And one thing is that we're trying to miniaturize the package by going from the kind of disks that you have now that spin to the solid state memory, for instance. We're trying to make it last longer, be smaller, cheaper, better in every possible way. And one of the things that I'm going to talk about is just the fact that we can configure these into arrays that track. So here's the thing as it exists, which is probably yay long, and hopefully I could come back next year and say it's only yay big. So now, one of the challenges of collecting a lot of acoustic data is that if you just take the data disks and you set them on a graduate student's desk, and you come back a year later, not much has happened. So we worked very hard to make graduate student hours more productive, which I know that sounds like an oxymoron in of itself. But and so mostly Sean Wiggins has developed this concept of a long-term spectral average. And what this means is that, so the spectra is sort of the basic tool of bioacoustic analysis, and it's looking at the frequency content versus time of some signal. And from this you discern a lot about the species that made the sound, and the other details about the behavior and that kind of thing. So long-term spectra says, if we're going to find these things, if we've got a year's worth of data and you can't have the analysts sit there for a year looking at it, we have to have a way of really rapidly finding what's of interest and then making some sort of assessment as to where to, maybe without even going any deeper, that what it is that took place. So the long-term spectral average is saying we can plot the spectra on any time scale that you want, and this particular time scale is 2 hours, but we could plot a day or a week or a few minutes. So in this particular case, those that spend hours staring at these things can read this kind of like a book in terms of, and I think this is from the Santa Barbara Channel, so there's a lot going on. But so for instance, when you see this kind of line of energy exactly at 50 kilohertz, there's a manufacturer of a sonar somewhere that tuned up, the transducer to be pinging at exactly that frequency. You see the same kind of thing here. So this is a human-made sound as opposed to a marine mammal sound. And you associate that with a bunch of noise down here, and if we take just a couple of seconds of data from this time right here, you can see, in fact, there's noise down here, which is a ship, and then something going ping, 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 at a regular rate. So there's just some kind of vessel going by with an echo sounder on, right? Now, and after you've looked at this for a while, you can look at that without having to go down to this and know what it is. Now, this guy is totally different because there's kind of this 10 to 15 to 20 kilohertz packet of energy, and then there is a shorter term packet of energy that goes almost all the way up to the top of the frame. And if we zoom in right here along the shoulder of it, at B, you can see all of these lines of energy, which are whistles of probably common dolphin. And if you stare really closely, there are some clicks in here. But even now, we can say that over the time span of approximately half an hour, a group of common dolphins swam by this instrument. Now, you may or may not need to know more about it, but just reading across this, we've said, okay, ship and dolphins, and there's another ship, and here's another ship, and on we go, and there are some more dolphins. Right? So it's a pretty rapid way, instead of sitting for 2 hours and listening to this. And in fact, you'd have to slow it down by a factor of 5 to hear it anyway. So this is a way of making the whole analysis go rapidly. This plot shows for the Southern California region where we've had deployments of these high-frequency acoustic recording packages. And there are some sites that have been occupied more than others. For instance, there are a couple of sites that we're trying to maintain a long time series for the Navy in terms of the level of sonar usage and the interaction of sonar with marine mammals related to the Navy's range. So, for instance, these two sites, and other sites get kind of moved around. But we've been trying to sample the environment, the California Bight environment, relatively broadly to get a sense of how marine mammals come and go, sort of geographically. But you can see it's a challenge because for acoustic instrumentation, you go out and you put it in somewhere and you hope it stays there until you come back. I mean, you hope that it doesn't move around. So we're getting excellent temporal coverage and relatively poor spatial coverage, right? Except, if you do this long enough, then you get your spatial coverage. So, and this is an example of the kind of analysis that the hardworking people in my lab do, and I get to enjoy looking at it after they've spent hours and hours putting it all together. So this is a plot of the detection of Adonoseed clicks, and it's plotted as time of day. And there's a shading here. I can't see it from this angle, but I hope you can, that shows when the sun is up or when the sun is down. And then this is a full year of data. So it's as if you sat out at this site, which is just south of San Clemente Island, and every minute you were telling me whether or not there were echolocating cetaceans nearby. And then there are little gaps every once in a while in the data. But you can see one of the things that comes out is right away is that most of the small cetaceans that occupy this site are nighttime foragers. Right? Here's an exception, but most of the time they're nighttime foraging, and they follow relatively well when the sun goes up and down. Now this is useful on some level, but it's an all, it's undifferentiated. And one thing that we can pull out at this point are the Cuvier's beak whales. And if you look at this now, you see a bunch of little dots, and it shows that this site it's quite common to have beak whales present at this site. And if you look at the pace of things, you can see sort of the pattern of the dives. And one thing we know about Cuvier's beak whale is they tend not to make echolocation clicks near the surface. They make the clicks at the bottom portion of their dives. And so this is some sort of proxy for potentially foraging dives of Cuvier's beak whale. And we learned, quite a bit about sort of the pace of that, and knowing more about our detection range will actually help us to turn this into a quantitative estimate of the density of animals around that site. So I think this is new, and this is something that we're going to learn a lot as we accumulate these kind of data. Now, I can learn how to use the pointer. What we'd really like is not just to say where the animals are present or absent, but to actually put a dot on the map that said, we heard an animal, and this is where it is in a three-dimensional space, both x, y, and depth. And so this is something that Sean's been working on for maybe the last year or so, and he calls it the tracking harp. I guess that would be a harp, or maybe we just drop the H and call it a tarp. But and what it is it has multiple sensors. Instead of a single sensor, it has 4 sensors, and in this particular one, they're configured as a little triangle with one sensor on top. And the idea is that by looking at the arrival of something like an echolocation click and all of these sensors, you can say the direction that the sound arrived at the instrument. Now, here's a result slide from that, actually the first deployment of the instrument. And the little triangle is where the instrument sits. This is just west of San Clemente Island. And he was able to track the foraging dives of 2 beaked whales that came down almost together. So there's the time, that's annotated along the track. And then, I don't know, this is a span of whatever it is, about a half hour. So you can see this guy comes down a few minutes before that guy, and they both move along the contours, over the span of about a half hour. I guess this guy who came down first goes back up sooner. This guy who came down second goes back up a little bit later. But they're probably more or less in some sort of social group and coordinating their behavior, listening to each other, clicking. But it's an interesting behavior to be moving along. They come down to the bottom or near the bottom, and they're moving along the contour, probably foraging, and then they come back up. So now, just by having this ability to locate individual clicks, we're getting behavioral information, about the detail of how these animals interact with the environment and potentially with each other. This is a slightly different view. These are common dolphins that are near the surface. Here's the same instrument. It doesn't look like the same contour, Sean, but and now the group of animals swims, must have been a different deployment, swim past the instrument over the span of sort of an hour and a half. And he's sort of done a centroid of the whole group. They're actually spread out in space, but you get a sense for sort of direction of travel and sort of how straight the travel is with time. In fact, I think this is a plot from...
It's just a
difference in the range.
Oh, is it?
Zoom
Okay.
out.
Yeah, I thought it was the same, but Kate Fraser, who's sitting in the room, made the plot, so you can ask her. Okay, so I was talking about one of the problems we've had with acoustics that makes it a mismatch to visual surveys is that often if we have a box here and we want to know what's in that box, then there are two ways to approaching it, and even in land surveys of things like birds, you can either follow some set of tracks, a line transect kind of survey, or you could go to some point and then do a survey around that point, or a point transect. And in fact, point transects are done for birds because you can go and settle down somewhere and let the birds adjust to your presence, and then you count around that spot, as opposed to walking through the woods and the birds are all scared off. Right? So the thing is, naturally, visual surveys can follow these sort of line transect, and visual surveys, at least in the ocean, are naturally not done as point transects, right? Starting and stopping the ship and hanging out. Now, you can do acoustic line transect by towing an array. And Jay and Barb and others in the room have made good use of that. But most of the other form of acoustics, the point transect acoustics, have really been sort of the forte of passive acoustic monitoring. But there's a new technology that's coming about that I think will change this, and that is these gliders and drifters. Because if you add acoustics onto a glider or a drifter, now you can convert it to where it's doing a line transect survey again, without the expense of having a ship present. So you get the same spatial coverage that you can get, you choose your whole box and cover it spatially. You can also, with things like the sea glider or the slocum that go up and down, you can collect the data at depth as opposed to being stuck at the surface. And it's much lower cost than having to conduct a shipboard survey. And potentially it's less disruptive to the animals because it's not making the sort of sound that you make with a big ship. So now this is something that's just coming online, and there was a test done, I'm going to say it was in early this month. And it was sponsored by ONR, and they went out to the Navy range west of San Clemente, and they put in every possible style of glider or drifter that they could find. And I didn't go on the test, but Sean did and took pictures of all of them so we could document this. But here are some drifters or vertical profilers that essentially they go up and down, but there's no control over how they move through the environment. And I guess there's a Woods Hole version and there's an Oregon State version in this. But basically, there's this package is basically a buoyancy package that moves the thing up and down in the water column, and then there's an acoustic sensor. And then there's the Slocum glider. Slocum is I think the Woods Hole version of the glider, where it has some sort of way of actually directing itself. It goes up and down vertically, but it can change its buoyancy in a way to orient so that once it gets to the surface, it has a position where it is and a position where it wants to be, and so during the next dive, it can move itself in that direction. So it's more of a vertical profiler, but with some horizontal motion. And so here is one of these Slocum gliders with the Demon, which is the new Mark Johnson recording. It's basically the tag, except made as an acoustic recorder, strapped onto the Slocum. And then Sea Glider is the University of Washington version of this, and I think it's been commercialized by a company called iRobot. So you could buy one of these, have an iRobot of your own. And here you see them along the side of the ship, and there's I think the acoustic sensor is strapped on sort of here, but again, they're vertical profilers, but can be directed. And then this is the glider on steroids made by Gerald Despain at Scripps, and it looks very much like some sort of stealth bomber, So, and now the thing that Gerald points out is that this is an actual glider in the sense that it has a more distance in the horizontal than in the vertical. In other words, going up and down, it'll be 10 to 1 in the horizontal versus its vertical motion, whereas the others are mostly vertical and less horizontal. And there are a whole series of sensors built in along the wingtip. There's an acoustic array built along both wingtips. And as part of this test, we invented a harp. I mean, this thing's big enough that it can take a harp and hardly notice it. So somewhere about in here is a harp. And there's a movie here, but as I remember, because of the Mac to PC transition, it won't play. But if I push it hard enough anyway, that's basically to show the deployment of this. This is the fantail of the sproul, and there's sort of a railroad track where they slide this thing back, and then it tilts up and slides off into the ocean, and away it goes. Okay, so the thing that my lab has been playing with is called a wave glider. And a wave glider has, and the wave glider has a surface float. The float actually always remains at the surface. And there's a dry box in the wave glider that we've introduced a complete harp, just like the kind of stuff that would go on the seafloor, except now it's at the sea surface. And then there's a tether from the wave glider that goes down to a submerged wing, and then from that wing, we're towing a little hydrophone. Now the wave glider, and so it's the same sort of parameters that you get for a harp. It could stay out for several months and record acoustic data. So this has the longevity to do an actual survey. You can lay out a track, and in fact, just as a test, the Liquid Robotics, the company that makes the wave glider, took a wave glider, put it in off of the big island of Hawaii. It came all on its own power over to San Diego. They looked at it, turned it around, and sent it back. It took a couple of months to get here, but they did a real track from here to San Diego. So we haven't had the nerve to do anything like that, but we might at some point. So how does this thing work? And it's one of these ideas that's so clever and so simple that you kick yourself after you realize it and wishing that you thought it up and that you held the 16 patents that these guys hold on it. But so the way it works is, if you think about wave motion, it's wave motion, the actual particle motion of a wave is sort of an elliptical motion near the surface. But that motion diminishes as you go down to depth. You know, it's basically, you know, relative to the wavelength, it's going to diminish exponentially. So if you take a surface float, this surface float is going to go up and down with this vertical excursion. And if you tie it with a tether to something that's in deeper water, the deeper water is more or less stationary. So now the surface float is pulling this wing up and down through this stationary deep water. Now the next bit is you make it a wing, except the wing is spring-loaded to be flat, but as you pull the float up, the wing takes a certain angle of attack, right? Now the fact that it takes an angle of attack, it's being pulled up this way, makes it move in a certain, you know, horizontally. Then you drop the thing down, the wing takes the opposite angle of attack, but because it's going down and has the opposite, you know, slant, it goes in the same direction. So each cycle is pushing it the same way. So then the last thing you have to add is a little rudder. It's a tiny little thing. I'm surprised they get away with such a small rudder, but that rudder, you can point which direction the wing is going off in. So you add an iridium satellite phone. You can talk to the thing anytime you want. It calls home once a minute, and a GPS receiver. So literally the one we have in my lab, which there it is in the service yard, and this one the company, for whatever reason, affectionately named this thing Tweety, because they don't usually make them yellow. But for an oceanographer, they decided to make it yellow so we could see it. That, by the way, most of these have been sold to the Navy, not for marine mammal purposes, as you might imagine, and not painted yellow, because they don't necessarily want people to see them. So what we do is, here's a little cart or whatever, they take it out to the end of the pier, set it in the water, actually use a small boat to drag it a little bit away from the pier. They pull a pin that separates the wing from the float. And then we go back into the lab, sit at your computer on an internet screen. You get a iridium message back from this that tells you where it is, and you say, okay, go here. And off it goes. So, as I said, we haven't had the nerve yet to do more than sort of simple missions around San Clemente and around the end of the pier. But at some point, for instance, we could run the calcopy lines and have this, constantly going back and forth, collecting not just acoustic data, but we could collect ADCP or, thermistor or whatever other kind of data that's appropriate. So I think this is going to revolutionize oceanography, not just for marine mammals, but for other things as well, because it gives us a persistent presence in the ocean that we don't have right now without having something expensive like a ship. So anyway, I think I've more or less used up my time. And coming to the session, I think it is really an impressive array of things that are going on. That the sort of things in the lab like genomics and biochemical assays, and then working with captive animals and also stranded animals, and then the new field techniques. All of these things together, we're learning a lot about marine mammals, and there are things that 10 years from now when we come back, I think we'll know a lot because of these advances in technology. Thank you.